apparatus or rest of the things will get duplicated in this phase, but chromosome will remain single. It won't replicate. It won't form its sister chromatid. Then it will enter the second phase, that is S phase. So what will happen in the S phase? Again here in S phase it will be single chromosome, like the single chromatid. I won't say chromosome, it will be a chromatid, single chromatid. So single chromatid, that is 23 pairs of chromosome, which means 46 chromosomes and 46 chromatids again, because it hasn't replicated yet. This S phase will take 9 hours to complete, and there will be DNA replication and chromosome duplication. So here in the S phase there will be chromosome duplication. That means this chromatid will form its sister chromatid, and this chromatid will form its sister chromatid. So here, as this is the process of replication, if you all can see, one chromatid is forming its replica, that is sister chromatid. So this is the S phase. Now from S phase, the cell will go into G2 phase. What is G2 phase? The single chromatid, that is we have 23 pairs of chromosome, 46 chromosomes, but here we have 92 chromatids. So if you all can recollect with me, we were having only single chromosome here, but it has replicated here. So now we have 92 chromatids, not 46 chromatids. This will take 4 hours to complete, and here the cell grows and prepares for mitosis and checks for errors. So now the cell is preparing itself to divide. So for dividing the cell, for dividing a single cell into two daughter cells, it will be needing the replication, that is its homologous chromosome or a chromatid we can say, to divide. So here in G2 phase, it will be having 92 chromatids. Now it will enter in M phase. M phase is nothing but mitosis. Now in mitosis, it will divide. So which will form two daughter cells. Each daughter cell will again have a single chromatid. Single chromatid, that is 23 pairs of chromosome, 46 chromosomes and 46 chromatids. Now we will be seeing M phase in detail. So far so good?
Yes.
Yes.
Hello?
Yes, good.
Yeah,
good.
you all with me?
Yeah.
Yes.
Yeah. So in M phase, now M phase has 12 phases: prophase, promophase, metaphase, anaphase, telophase. First is the prophase. So now here what we are seeing is a daughter cell which has grown and which has duplicated its chromosome content and rest of the things. So this is the daughter cell. It is yet to divide. This is this cell which is yet to divide. This is the phase that we are seeing here. So now, as I have said before, I have mentioned before, that there are some organelles that are called centrosomes. They are present in the plasma. So these centrosomes have microtubules. These are extensions. These are spindle-shaped extensions. Apart from that, what we have is the nucleus and the chromosomes with a centromere and attached kinetochore. Prophase, what happens? Chromosomal condensation occurs. Apart from that, cytoskeleton disintegrates. So cytoskeleton is nothing but the nuclear envelope. So this will disintegrate. This will break up. The nuclear envelope will break up. There will be spindle formation between centromeres. So spindle formation is nothing but the centrosomes forms the extension. Those are called as spindle formation. And then this will proceed to prometaphase. So prometaphase, metaphase are like one and the same, but they are again divided into two. So what happens in these two phases is, as the nuclear envelope is disintegrated, chromosomes are free to move around in the cytoplasm. So chromosomes get dispersed in the cytoplasm. What we will do is, they will go and attach the centromere. Centromere is center of the chromosome, that is kinetochore. So they will go and attach to the kinetochore. like what they have shown in the picture. So chromosomal microtubules attach to the kinetochore of the chromosome. Chromosomes move to the spindle equator. What is spindle equator? Is the center of the cell. So all these chromosomes will be moving in the center of the cell, and the microtubules or the centrosomes move further apart in the extremities of the cell. So these are centrosomes. They are sending microtubules that are getting attached to the center of the chromosome, and chromosomes are aligned in the center of the cell. This center of the cell is also called as metaphase plate. So chromosomes are aligned along the metaphase plate attached by the chromosomal microtubules to both the poles. Okay, so now, hello?
Yes.
Is there any problem?
No problem. No problem.
Okay, yeah. So now what will happen is, in anaphase, the centromere splits and chromatids get separated. So the centromere is the center which is holding the chromosome together. So it splits, and these chromatids, the sister chromatids that I have said before, they will get separated. And chromosomes move to the opposite spindle poles. As we have seen, the centrosomes are on the opposite side. They will pull the chromosomes towards them with these spindles, microtubules, and they will move apart. The chromosomes will move apart, and these spindle poles will further move apart. So there will be moving apart of both the things. And in the telophase, or the main phase where it actually gets divided, it causes further moving apart of the chromosome, and all the procedures that has happened here will get reversed. So what will happen? The nuclear envelope has disintegrated. Here it will form again. It will start forming around each of the centrosome. Like we can see here, the nuclear envelope is starting to form. Here also it is getting formed. Microtubules are getting apart. The chromosomes are near the centrosomes, and basically the cell is preparing itself to divide into two daughter cells. Like each daughter cell will have a single chromatid. So chromosomes clustering at opposite spindle poles, nuclear envelope assembles, organelles reform, spindle dissembles and reassembles as the cytoskeleton in the two new cells. So now the cell has prepared itself for the final division. So what does this final division known as? It is known as cytokinesis. So cytokinesis is nothing but the main division of the two daughter cells. So as we have seen, they have moved apart. Now in between, like here, in between, there will be a contractile ring. This contractile ring will divide the two cells. Now each daughter cell will have 46 chromosomes and 46 chromatids. So 92 chromatids that we have already seen gets divided and forms again into 46 chromatids. So far good?
Yes.
Yeah.
Okay. So now these like what the cell cycle is just the superficial picture of the cell cycle. What are the main factors that causes cell cycle are nothing but the cyclin dependent kinases. These are the main heroes of the cell cycle, we can say. So cyclin dependent kinases are like proteins which are dependent on the cyclins to function. So there are CDKs and cyclins. So both of these complexes help together to proceed the cell to further into cell cycle. So in G1 phase, we have cyclin D and CDK4/6. In G1S phase transition, we have cyclin E CDK2. In S phase, we have cyclin A CDK2. In G2 phase, we have cyclin A CDK1, and in M phase, we have cyclin B CDK1. So these are like you have to remember these cyclins and their components. So these factors help cell to proceed into cell cycle, and there are the different modes of regulations through which these factors operate. So first is periodic transcription of gene, second is phosphorylation, third is action of inhibitory proteins, and fourth is nuclear import or export. So what is periodic transcription of gene is? There is something known as repressor complex. Now this is very Like this, you have to remember it. Like you don't have anything else to do. You have to remember these things. So there is something known as repressor complex, which binds with CERM protein, and this binding causes inhibition of cyclin E. Now CDK4 and 6, what they do is they causes phosphorylation of this binding, which further removes the inhibition from cyclin E and makes cyclin E available so that the cell can proceed from G1 to S phase. So this is one of the mode of regulation how CDK4 and 6 helps in proceeding with the G1 to S phase. Second is CHR. CHR is also a repressing element for cyclin B and A, which on phosphorylation provides cyclin B and A for further procedure to go. Then there is something known as phosphorylation. Now cyclin and CDK are with the T loop. They are in the inactive state. So when phosphorylation of T loop occurs, there is an active form of cyclin CDK complex. Now again, if the phosphorylation occurs here, then they again gets into an inactive stage. Or if there are any inhibitors like CKI, they will again go into an inactive stage. So basically, phosphorylation is one of the mode where it will depend whether the cyclin and CDK are going to be in the active form or they are going to be in the inactive form. So whenever there is whenever the cell is in danger, or when the body or the cell does not wants to proceed itself to further stages, then there is the phosphorylation at the cyclin level, and cyclins then regulate it to restrict it to the S stage, or we can say G1 stage, and to further stop it from going in the further stages. Then there is something known as inhibitory proteins. So there is INK4 family, which is P5, P15, P16, P18 and P19. They help inhibit CDK4 and CDK6. Then there are CYP family, which are inhibitors of CDK2 and CDK1. Then there is PRB, that is protein retinoblastoma, which inhibits cyclin E and cyclin A along with CDK2. So these are the inhibitory proteins which again regulate cyclins and their availability for the cell cycle to proceed. Then there is basic mechanism, nuclear import and export. So whenever the cell wants itself to get restricted, it won't import the cyclin B CDK1 complex. So it will keep the cyclin B CDK1 complex out of the nucleus so that it won't proceed further to the further stages of the cell cycle.
Hello.
Then, yeah.
Please, can you go to the last slide?
This one?
Just one before that.
Okay, yeah.
Here, what we can summarize is that CDK4/6 phosphorylation is proceeding the cycle, and cyclin phosphorylation is inhibiting. Yeah, that we.
No. See, cyclin and CDK both are required for a cell to proceed further. Okay. So whenever it is what we can say is it is like positive inhibition or a negative inhibition. If the cell feels, or if the body feels that there is danger for this cell to proceed, like if there is a cancer cell and we don't want the cell to proceed further, so what the cyclins or rest of the inhibitors will do is they will inactivate the cyclin and CDK complex. So if cyclin and CDK complex are not available, cell won't proceed further. Cell will be restricted to where it is, and it will undergo repair, or maybe it will undergo arrest. It is the body's saving mechanism.
Cyclin will inhibit CDK4. Yeah.
Sorry?
Cyclin will inhibit CDK, or what? CDK inhibitor?
No, cyclin won't inhibit. There are certain inhibitors.
Okay,
like
that
these.
I can. Okay.
Yeah. These will inhibit. And cyclin and these complexes, if they undergo phosphorylation, then they will go into an inactive stage.
Okay. So phosphorylation, as we can see in the upper diagram, that CDK4/6 phosphorylation is activating now. So
This
this is
CDK4, see here it's a repressor complex. This
okay.
E2F4, P10, P107 histone, these are repressor of cyclin E. So basically what CDK4 and 6 are doing is they are phosphorylating these, they are repressing further repressor of cyclin E and they are making cyclin E available.
Negative becoming positive.
Yeah, exactly.
Okay.
So CDK4 is in G1 phase, right? And for G1 phase to proceed to S phase, we need cyclin E. So basically CDK4 are making cyclin E available by removing their inhibition, which is done by repressor complex.
In phosphorylation, second photo, T loop को कौन phosphorylate कर रहा है? जो written as cyclin will attach CDK with T loop. Inactive stage में है phosphorylation of T loop. So वो complex को T loop को कौन phosphorylate कर रहा है? Which substance is coming and phosphorylating that inactive complex and making it active?
Phosphorylation कौन कर नहीं, phosphorylation is basically वो phosphorylate हो रहे हैं by some component. Negative regulatory phosphorylation is what they have mentioned. They haven't given anything like who is phosphorylating them.
Okay.
Yeah. So basically एक बार phosphorylation होने के बाद they are going into an active stage, but वापस
Active
phosphorylate
stage.
अगर हो गए तो inactive stage में जा रहे. So it is a reversible. This procedure is reversible. If एक वो जो phosphorylation है वो remove हो गया तो again वो active हो जाएंगे. So this will undergo, this procedure will undergo depending on what need is here in this cycle. Whether we really want CDK cyclin complex or not, depending on that, this will undergo, whether it has to be in the active or inactive stage.
So like T loop phosphorylation is inhibiting, that I can understand. T loop phosphorylation is inhibitory. That na?
Yeah.
Okay.
इसका मतलब है और T loop को अगर phosphorylate नहीं किया तो inhibitory state में cell cycle not further proceed.
Yeah, right. बेसिकली यहाँ पे साइक्लिन्स पे ही पूरा सेल साइकिल चल रहा है, साइक्लिन्स एंड सीडीके. अगर साइक्लिन्स एंड सीडीके ही अवेलेबल नहीं है तो सेल साइकिल प्रोसीड नहीं होगी.
Okay. Thank you.
And these are the different modes how cyclins are controlled or cyclins or CDK are kept in check. So these are different modes that I have told: periodic transcription of gene, phosphorylation, action of inhibitory proteins, nuclear import and export. ये सब उसके तरीके हैं कि साइक्लिन्स और सीडीके को अवेलेबल करना है या नहीं करना है उस वक्त पे.
Okay.
या सो अभी तक सब ठीक है. शुड वी प्रोसीड?
Yes.
Okay. So now induction of cell cycle phase transition. So now the two important phases that we can make out is G1 to S phase ka transition is important one, and another important one is G2 to M phase ka transition, because S phase will be the synthetic phase, M phase will be the mitotic phase. So before these two important steps, there should be some checkpoints, whether the cell is good to go ahead or not. So these two are the checkpoints, basically, we can say. Now what happens here is, as we have seen, in G1 and S phase, we require cyclin D, CDK4, cyclin E, cyclin A. Along with that, we require CDK2 and CDK1, right? So these CDK2, CDK1 are kept inhibited by CIP-KIP family. So as I have already mentioned, these are inhibitory proteins, CIP and KIP, which inhibit CDK2 and CDK1. So here in the image, you can see the blue thing, that is CIP, KIP family. They have inhibited CDK2 and CDK1. So without CDK2 and CDK1, cyclin EA won't be able to do their work. So now what happens is, whenever there is, the cyclin E and A will get just accumulated. They won't be able to function unless they get their partner CDK2, right? So with the accumulation of cyclin, if internal and external environment are permissive for proliferation, like the body, like the cell knows now the G1 phase has cell has undergone G1 phase, so cell abhi अच्छा है आगे proceed होने के लिए. So both internal and external environments are okay. Cyclins जब accumulate हो जाएंगे, they will titrate these inhibitors. So जैसे अगर शक्कर मिलाया पानी में तो शक्कर पूरा डिसॉल्व हो जाएगा बिकॉज पानी ज्यादा है. तो वैसे ही साइक्लिन ई जो है या साइक्लिन ए जो है वो ज्यादा एक्यूमुलेट होने के बाद दे विल टाइट्रेट द इनहिबिटर्स व्हिच आर CIP KIP फैमिली. सो वंस दे टाइट्रेट दिस इनहिबिटर्स, अपने आप वो जो इनहिबिशन है वो हट जाएगा. सो CDK2 विल बी अवेलेबल एंड CDK1 विल बी अवेलेबल. सो नाउ व्हाट साइक्लिन्स विल डू, cyclin A and CDK complexes will further phosphorylate these inhibitors. Like they will further dissolve these inhibitors. And these inhibitors will undergo ubiquitin mediated proteolysis. So basically, they will remove all the inhibitors. So this is one of the step that is carried out at this point. That is this restriction point, we can say. If the internal and external environments are not permissive, there won't be any accumulation of cyclin and these procedures won't happen. So the cell will remain restricted here only. Okay. Now what happens at G2 and M phase is we require cyclin B, and along with cyclin B there is CDK1. So this is kept inhibited by negative regulatory phosphorylation at threonine and tyrosine. So now this is inhibited. So cyclin B के लिए CDK1 available नहीं है अभी. So what will happen is CDC25B and CDC25C will cause dephosphorylation of these phosphorylated threonine and tyrosine. Once they are dephosphorylated, it will be available for cell to make cell undergo further procedures. Like cyclin B and CDK1 will be available and they will help cell to proceed to M phase. So these are the two transitions where these things are kept in check. Now transition within M phase. Now as we have seen before, as we have seen before, metaphase and anaphase. In between metaphase and anaphase, in metaphase the chromosomes are at the metaphase plate in the center of the cell. In anaphase they get separates with the microtubules. So in between these again there is a checkpoint, we can say. That is whenever the kinetochores are attached to the spindles, that is these spindles get attached to the kinetochores. This is this step. So kinetochores attached to the spindle. This causes positive, what we can say. This causes stimulation of CDC20. Now what CDC20 will trigger APC and C. And what APCC will do? They will inhibit securin. And the securin separase complex will be lost. Now once the securin and separase complex will be lost, the separase will help to cleave the adhesion between the two chromosomes or the two chromatids, and then the two sister chromatids will get separated. So this is the basic procedure of chromatid separation, how it happens. So CDC20 will stimulate APCC. APCC will separate these two, that is securin and separase. Now securin is inhibiting separase here, we can say. But once it is separated from the separase, separase will come and do its work and it will further separate the sister chromatids. So this is one of the step where the cell is kept in check, like before proceeding to anaphase. Now another thing is quiescence and differentiation. So quiescence is nothing but if the state of the cell cycle exit is reversible, it is called as quiescence. That is whether like if the mitogens or growth factors and hormones are like good enough, the cell will undergo a further differentiation. If not, then the cell will exit. But again, if they are available, cell will again come and go further into its cell cycle phase. So this is quiescence, which is reversible. Differentiation is nothing but cell will convert itself into a different cell form. Like one cell, we can say that cancer cell, when cancer cell is normal or it is into a mild dysplasia or moderate dysplasia, that is nothing but a differentiation. So what happens in quiescence is there is upregulation of CDK4, 6 and CDK2, which causes accumulation of cell in G1 and then the cell exits. Then there is downregulation of CDKs and cyclins at transcription level, which further causes cell to exit, and there is reduced rate of protein synthesis. So when these factors are not available, these factors will cause the cell to exit itself into a arrest form. But when these factors are again available, the cell will again re-enter into the cell cycle form. Another aspect that is kept in check are the inhibitory proteins, as we have already seen, INK4 group, then CYP group. These proteins will keep inhibitions over these cyclins, and these cyclins will further inhibit the cell to proceed into the cell cycle, and then cell will go into a cell arrest. Then there is something called as checkpoints. So checkpoints are basically at 3 levels, that is G1, then there is S checkpoint, and there is G2 checkpoint. Now first we will be seeing a G1 checkpoint. So how this occurs is when the cells are constantly faced with insults, whether it is internal or external, resulting in a damage that can threaten their survival. To repair these damages, various mechanisms have evolved, and these are nothing but the cell cycle checkpoint. So जब सेल को कोई खतरा होता है, तो सेल को फर्दर प्रोसीड होने से बचाने के लिए ये सब चेक पॉइंट्स रहते हैं. So there is replication checkpoint, DNA damage checkpoint and spindle integrity checkpoint. So यहाँ पे G1 arrest पे क्या होता है, like CYP inhibitors जो है, they keep cell arrested at G1 as we have seen before, because वो दोनों जो है वो inhibit करते हैं CDK4, 6 and CDK2 को, CYP inhibitors. But जब यहाँ पे any damage is seen, like सेल को कोई डैमेज होता है, तो ATM एंड ATR प्रोटीन काइनेजेस एक्टिवेट हो जाते हैं, और वो सेंसर्स रहते हैं DNA डैमेज के लिए. So when these proteins are activated, they activate P53. P53 will further activate CYP inhibitors, or will further bring into picture the CYP inhibitors. And CYP inhibitors will further inhibit CDK2, CDK4. अब ये दोनों जब inhibit हो गए हैं, तो सेल कैनॉट प्रोसीड फर्दर टू एस फेज. सो वो सेल यहाँ पे ही अरेस्ट हो जाएगा. सो इट इज नथिंग बट अ G1 अरेस्ट. Another thing is whenever P53 is activated, there is another inhibitor that comes into action is GADD45. ये inhibitor क्या करेगा? It will inhibit CDK1. So CDK1 यहाँ पे required होता है to proceed cell into M phase. तो वो नहीं होगा, वो नहीं रहेगा तो साइक्लिन बी भी नहीं कुछ काम कर पाएगा, तो सेल यहाँ पे ही अरेस्ट हो जाएगा. दैट इज कॉल्ड एज G2 अरेस्ट. नाउ दीज आर P53 डिपेंडेंट वी कैन से इनहिबिटर्स, दोनों भी. देयर इज समथिंग व्हिच आर P53 इंडिपेंडेंट. सो व्हाट आर दोज आर CHK1 एंड CHK2. CHK1 और CHK2 क्या करेंगे? दे विल डायरेक्टली इनहिबिट द सेल, या फिर दे विल इनहिबिट CDC25B एंड CDC25C, विच इज कॉजिंग डीफॉस्फोराइलेशन, जो अपन ने यहाँ पे देखा था. यहाँ पे CDC25B और CDC25C क्या कर रहे हैं? डीफॉस्फोराइलेट कर रहे हैं इसको, CDK1 को, जो कि ऑलरेडी इनहिबिटेड है, बट डीफॉस्फोराइलेशन के बाद वो अवेलेबल हो रहा है. सो ये स्टेप को ये लोग रोकेंगे. CHK1 एंड CHK2 दे विल स्टॉप दिस स्टेप. सो अगर ये ही नहीं हो रहा है, तो CDK1 और CDK2 भी अवेलेबल नहीं होंगे, एंड देन सेल विल गेट अरेस्टेड S फेज एंड G2 फेज. ये समझा क्या सबको दिस स्टेप?
हाँ समझ आया.
Yes, yes.
Okay. Now another is spindle integrity checkpoint. The mechanism of delay at prometaphase or metaphase in response to spindle defects or improper chromosome attachment. So अभी जैसे we had seen before, अगर kinetochores जब spindle के साथ attached होते हैं तो ये further procedure होता है सारा and वो separate हो जाते हैं. पर अगर इनका attachment जब ठीक नहीं है तो there is something damage that has caused and that's why the attachment is not proper. So cell को पता है कि अब divide होके गलत होने से अच्छा it is better to not to divide. So what whenever this attachment is not proper, what they will do that they won't activate CDC20. ये step ही नहीं होगा जो कि पहले हुआ था. जब attachment ही proper नहीं है ये inhibit रहेगा. Further APC-C will be inhibited which will further inhibit into a homozygous one or to lose its heterozygosity. So उसमें जो लेटेंसी पीरियड रहता है कैंसर होने का वो बहुत ही कम रहता है. इसमें लेटेंसी पीरियड बहुत ज्यादा रहता है बिकॉज एक बार होने से नहीं होता, वापस हुआ तो होता है. तो इसमें लेटेंसी पीरियड बढ़ जाता है. इसमें इमीडिएटली हो सकता है कैंसर बिकॉज एक ही बार होने से तुरंत हो जाता है. सो दिस इज 2 हिट मॉडल जो कि मेन और वी कैन से द कार्सिनोजेनेसिस का इम्पोर्टेंट एस्पेक्ट है. नाउ म्यूटेशंस जो होते हैं दे आर डिफरेंट काइंड्स. सो एज वी हैव सीन जो डीएनए रहता है उसमें वी नो दैट डीएनए इज मेड अप ऑफ गुआनिन, साइटोसिन, एडिजिन एंड थायमिन. सो अगर एक सिंगल पॉइंट पे म्यूटेटेड हो रहा है लाइक साइटोसिन पे हुआ तो इट इज कॉल्ड एज पॉइंट म्यूटेशन. या फिर अगर ग्रुप पे हुआ 2-3 ग्रुप पे हुआ तो इट इज कॉल्ड एज डीएनए एम्प्लीफिकेशन. So these are the two kind of mutation that occurs. Apart from that, there is something known as epigenetic modifications. So what are these? That is histone modification or DNA methylation. So जो histones रहते हैं जो DNA को रैप करते हैं उनको tails रहते हैं. तो उसमें acetylation होता है या फिर मिथाइलेशन होता है. अब अगर acetylation हुआ जैसे कि यहाँ पे acetyl group अटैच हो रहा है तो उसमें एक्टिव क्रोमैटिन ट्रांसक्रिप्शन होगा. That is one kind of mutation. अगर मिथाइलेशन हुआ तो रिप्रेस्ड क्रोमैटिन ट्रांसक्रिप्शन होगा. तो अगर रिप्रेस हुआ है तो आगे के कोई भी जो स्टेप्स है उसके वो नहीं होंगे. एक्टिव हुआ तो आगे के स्टेप्स ज्यादा होंगे. सो दिस इज वन काइंड ऑफ मॉडिफिकेशन हिस्टोन में जो कि म्यूटेशन है. अनदर वन इज मिथाइलेशन, डीएनए मिथाइलेशन. सो डीएनए को जब मिथाइल ग्रुप अटैच होता है तो इट इज एसोसिएटेड विद जीन साइलेंसिंग. इसमें दिस इज ऑल्सो वन काइंड ऑफ म्यूटेशन टू कन्वर्ट अ सेल इनटू अ कैंसर सेल. नाउ अनदर वन इज प्रोटोऑन्कोजीन जो विच आई हैड ऑलरेडी मेंशन्ड, आरएएस जीन. सो आरएएस जीन हर एक सेल में रहता है. known as growth factor receptor. So whenever there is growth factor which get attached to the receptor, it activates this RAS gene. Now when RAS gene is activated, it undergoes phosphorylation. Multiple phosphorylation will occur and it will causes activation of transcription factor. So DNA पे भी एक जीन रहता है तो वो एक्टिवेट हो जाता है when it gets phosphorylated. growth factors आके उसको स्टिमुलेट करके फॉस्फोराइलेट करके फिर यहाँ पे एक्टिवेट करते हैं. नाउ ये जब एक्टिवेट होता है इट विल कॉज प्रोटीन्स, इट विल फॉर्म प्रोटीन्स फॉर सेल ग्रोथ विच आर G1 एस्पेस जो सीडीके और साइक्लिन प्रोटीन्स है ये आरएएस जीन प्रोवाइड करता है. तो एंड अनदर जीन इज MYC जीन विच हेल्प्स इन सेल ग्रोथ, सर्वाइवल एंड एक्टिविटी. बट व्हेन दीज टू जीन्स आर म्यूटेटेड, अब आरएएस जीन अगर म्यूटेटेड है तो ऑब्वियसली ज्यादा प्रोटीन्स देगा वो. जब ये ज्यादा वो ये जब स्टिमुलेट होगा तो ये म्यूटेटेड जीन सब कुछ ज्यादा प्रोड्यूस करेगा, प्रोटीन्स भी ज्यादा देगा. तो सीडीके और साइक्लिन्स भी ज्यादा आएंगे. अब जब सीडीके साइक्लिन्स ज्यादा आएंगे तो सेल का जो ग्रोथ एंड रेप्लिकेशन है वो भी बहुत ज्यादा हो जाएगा विच इज द बेसिक ऑफ कैंसर, विच इज द एक्सेसिव प्रोलिफरेशन वी कॉल. तो कैंसर जो बढ़ता है दिस इज बिकॉज ऑफ दीज थिंग्स, म्यूटेशन इन आरएस जीन, अनदर इज म्यूटेशन इन MYC जीन. अब MYC जीन अगर म्यूटेटेड है तो ऑब्वियसली सेल ग्रोथ और जो सेल सर्वाइवल है वो बहुत ज्यादा होगा. इसलिए कैंसर सेल्स में जो ग्रोथ रहता है बहुत ज्यादा और ड्रास्टिक ग्रोथ, वी कैन से ड्रास्टिक प्रोलिफरेशन, it is because of the mutation in these two genes. Then tumor suppressor genes में क्या होता है? So tumor suppressor genes में क्या होता है कि whenever there is a damaged DNA, P53 will get activated, which acts as a transcription factor. Now P53, what it will do is it will cause cell arrest, cell repair and cell apoptosis. अब normally जब P53 activate हो जाता है और उसको पता है जबकि डीएनए डैमेज है तो अपने को प्रोसीड नहीं करना है डैमेज डीएनए को इनटू अ सेल साइकिल. सो इट विल कॉज ऑल दीज थिंग्स. सो इट विल स्टॉप प्रोटीन प्रोवाइडिंग प्रोटीन्स एंड विच विल कॉज सेल अरेस्ट. It will repair whatever the damage that has caused. So it will provide proteins that are important for cell repair. Or it will cause apoptosis, that cell will undergo a cell death, natural cell death. But what if these P53, that is tumor suppressor gene, is mutated? So inactivation of tumor suppressor gene or mutation of tumor suppressor gene won't cause these things to occur. That is cell arrest नहीं होगा, cell repair भी नहीं होगा, cell apoptosis भी नहीं होगा. So जो damaged cell है वो continue करेगा to grow and to proliferate, which is the basis of cancer. So this was all about cell cycle, basic of cell cycle. इसमें जो head neck specific है वो मैं next lecture में लूंगी, because it will be like more in a single lecture, more to imbibe.
Next Sunday your camp is not there, na?
Yeah, mostly नहीं रहेगा. रहा तो भी then Saturday I will do it if you all are free.
These people are also talking about Saturday actually.
You all let me know. वैसे मैं ले लूंगी, कुछ इशू नहीं है. ये क्लियर हुआ है ना दिस चैप्टर?
हाँ
nice, thank
बहुत अच्छे से यार.
जोई excellent presentation. Thank you for the presentation.
Yeah. You just like it will help you more if you all go and read it once from Devita.
Can you please share this?
So this everything is from Devita.
Yeah. This all chapter is from Devita. I guess it is 6th edition. I don't know the edition. I feel it is 6th edition.
अभी 11th edition चल रहा है रे. 12th चल रहा है.
11th edition चल रहा है.
12th चल रहा है.
No, but cell cycle is very basic. Like you can read it from any edition, I feel.
नहीं पूछेगा whether it is 6th or 10th edition, एक क्वेश्चन पूछेगा.
Sorry.
6th edition और 12th edition कुछ भी होने दे, क्वेश्चन पूछेगा एग्जाम में. He will ask.
हाँ, right. ये 8th edition का है.
Basic है.
It is too old. 8th is like recent one.
चलेगा यार.
ठीक है, मैं PPT शेयर करती हूँ. It is more of picture. I don't know you will be able to understand it or not, but then at least when you read the chapter, then you will get
Revision
to
के लिए help
recollect
हो
it.
जाएगा.
हाँ, ठीक है. Okay. Yeah.
Anybody
Thank you,
else going to present क्या? कहाँ गया? मोहित was going to present. Who was going to present?
Yeah. Knudsen two-hit hypothesis it is.
मुझे याद आ गया तो मैंने लिख दिया. I was
Yeah,
not.
right.
मोहित प्रेजेंट कर दिया ऑलरेडी.
कर दिया क्या? वो एक बोल रहा था ना कि मेरे को राउंड्स पे
हाँ.
जाना है, मैं प्रेजेंट करूँ क्या?
करके चलेगा शायद. वो मोहित ही था. ओरल मेट्रोनोमिक की मतलब.
स्वप्निल पूछ रहा था शायद.
चलो पूछते हैं ग्रुप पे. थैंक यू जूही.